My name is Nico Philipp. I'm a finishing up third year PhD candidate here at KU in the Jayhawk Athletic Performance Lab. Talk a little bit about my poster here. We're here at the Tsai Human Performance Alliance meeting symposium. My poster is kind of unique in the way that it came through a between institution collaboration. That's why I'm presenting it here. So through some Alliance funds, I was able to go out to the University of Oregon last spring for about 2 to 3 weeks. Got to collaborate with folks out there. One of the research interests that I have is studying the biomechanics of horizontal deceleration in team sport athletes. That's something that the folks in the athletic department at Oregon were particularly interested in. They had a lot of data on a deceleration test that they've performed with their football athletes, some other athletes. However, through discussions while I was out there, we identified some limitations in the way that data is quantified. So with this project here, what we did is we were interested in studying the between-rater agreement, so reliability for studying horizontal deceleration using radar technology, so instantaneous velocity from a radar device. So with this project, we had 4 different raters that manually processed the instantaneous velocity data. One researcher made all the calculations, and then we ran comparisons between all the different raters to see if the agreement, if it's not there, because in a perfect world we would want to have good agreement between raters, and if there is no good agreement, we'd want to have one designated person to process the data. What we found was that there was pretty decent agreement between raters. However, with instantaneous velocity data, we want to make sure we are spot on. So if possible, one person processes the instantaneous velocity data, from which we then later calculate deceleration metrics. One unique thing was that this was done on actual athletes, so we had a sample of 90, which is very rare in athletics. I think we had about 75 of Oregon football players, and the rest was lacrosse athletes, so we had a pretty good sample size of a highly trained population, very applied, and yeah, this is what my poster is about.
I'm Eserhaut. I'm a third-year PhD student in the Athletic Performance Lab, and I do a lot of our endocrinology work. My dissertation stuff is focused on the application of blood flow restricted resistance exercise to resistance trained individuals. So this is the first study in a series of 2 that we'll be doing on the application of BFR to this well-trained cohort. This study, we brought in 18 well-resistance trained men and exposed them to a passive BFR condition, which was just the occlusion stressor without exercise. Then they came back in and with the same occlusion pressure, had them do 4 sets of seated leg extensions to muscle failure. And then the final visit was just the resistance exercise stressor, where they matched the repetition performance without any BFR stimulus whatsoever. So we sort of took blood flow restricted resistance exercise and broke it down into occlusion only and exercise only. The outcome variables here were primarily on the endocrine side and metabolic side. So we measured blood lactate with some finger stick blood sampling, salivary cortisol with salivary analyses, and then an enzyme called salivary alpha amylase, which is related to the sympathetic fight or flight response. So we can see the application of blood flow restricted resistance exercise does significantly amplify the blood lactate response in well-trained men, the salivary cortisol response in well-trained men, and then also the salivary alpha amylase response. So it seems like the fight or flight response during BFR is significantly elevated versus a workload match condition and is greater than just the occlusion stress in and of itself.
Hi, I'm Jessica Provost. I'm a registered dietitian and sports dietitian, and so I get to work in the Jayhawk Athletic Performance Laboratory to bring nutrition information and awareness into our sport research, because a lot of our projects do need to have nutrition controls so that we can really assess performance. My project was based on using a measurement of energy availability, because in athletes, when they're not eating enough, they can have negative performance effects. And so looking if we could possibly use this kind of trick where we use their metabolic rate to predict whether they're at risk of not having eaten enough, and see if we can assess that just with this other test that's not very specific and not nearly as involved as asking them every single thing they ate or every single activity they did.
Hi everyone, my name is Minh. My project is about position-specific conditioning effects on female soccer athlete at a collegiate level. So basically I test how athletes do conditioning with the balls will be different from doing conditioning with high-intensity interval running or small-sided games as a normal type of conditioning for a female soccer player. And we test this project on 18 different soccer players at the Division II level, female soccer level. And interestingly, we find that compared to a normal conditioning type of small side game and high intensity running, our endurance tests improve the same. However, the technical skill test of the Loughborough soccer passing test, our athletes that participate in conditioning with the ball actually improving skill while maintaining the same amount of training load throughout the 12 weeks intervention program. And that leading to the conclusion that position-specific conditioning, PSCT, can actually improve skill for this athlete group and also maintain conditioning level comparable with other conditioning type, with also maintain similar training load across methodologies.